functions of membrane proteins. The lipids in the membrane in the plasma membrane of cells, they are very much similar of different cells. But the assortment of the proteins, that is very much different, and that has great influence on what the plasma membrane is doing and what the cell is doing. They are, for example, the channels. We are taking specific substances in or out through a hole. Most of these channels are selective. They are taking just one specific type of substance in. Therefore, many cells have different ones of these channels, selective channels, so that we can take different one for each one, but selective in particular this one in. Then there are transporters. They are taking a substance and then they are shapeshifting and transporting it in, like a snake. They're taking it and when like a snake, they're bringing it in. For example, amino acids needed to synthesize proteins in the human body come through that way into a cell. Then there are receptors. There's a specific ligand called substance stuff, and that thing fits exactly therein. So having a specific shape or something, or somehow it works, it's not directly said how, that specifically this type of substance, this ligand, can dock onto it. And when it had docked there, it changes when the outside of a cell. It's not something differently because something else is now there. For example, antidiuretic things are docking in the kidneys that way on and changing their permeability. And then there are other things. There are then the cell identity markers, and there are glycolipids and glycoproteins, and for example, a major class of them are the major histocompatibility, and we are looking if a cell is of the same body as they are or of a different one, so we're checking for foreign ones. And then there are enzymes, for example, in the small intestine, there can be lactase in it, and if it comes now drunk for milk, disaccharide lactose, they can take something in it, making a reaction and splitting it out again. So splitting up this disaccharide lactose in the small intestine. And whether which phase they are pointing these enzymes, where they are doing that thing, that job, that reaction. And then there are linkers, and they are taking a filament on the inside or outside, and that gives stability and structure to a cell, and also can help with movement, and they can also link with them to other cells. And that is according to Gerard Tatoga and Senegabowski's Principles of Anatomy and Physiology book on the pages 63 and 64, but this is no substitute for medical knowledge and also no medical